Hello everyone, my name is Krishna Jayaram and I'm a current sophomore here at Lehigh University studying bioengineering. My research is under Professor Tomas Gonzalez Fernandez and it focuses on recapitulating the effects of the osteoarthritis inflammatory environment on mesenchymal stem cell chondrogenesis. Osteoarthritis is a degenerative disease involving damage to articular cartilage in the knee joint with several risk factors including age, gender, and obesity. However, the most predominant risk factor that we are investigating is the degradation of extracellular matrix, which leads to inflammation. This can be regulated and modeled by pro-inflammatory M1 macrophages in a laboratory setting. The objective of this study is to explore how inflammation affects human mesenchymal stem cell chondrogenesis by incorporating cell pellets in macrophage-conditioned media. In terms of the bigger picture, in order to treat osteoarthritis with anti-inflammatory therapies, we must develop a robust model of inflammation for our in vitro studies, which is what this smaller study aims to do on a larger scale. Moving on to the experimental design of this study, it will take place over the span of 4 weeks. During the first couple of days, our cells will be seeded into expansion media to be grown and proliferated, and then for 3 weeks they will be allowed to differentiate, and then in the 4th week they will be split into the different experimental groups that are outlined in this figure. In part A of this figure, we can see that incorporating mesenchymal stem cells into chondrogenic media allows them to differentiate into chondrocytes. This is because chondrogenic media contains chemically defined media, insulin transferrin selenium, dexamethasone, transforming growth factor beta, linoleic acid, and ascorbic acid. All of these factors together is what allows for the chondrogenic differentiation. We will be using this chondrogenic media as a baseline in our study because it is an indicator of cell growth and differentiation, and we will be able to compare the other experimental groups against this group. Moving on to part B of this figure, we can see two different groups, which are RPMI and RPMI along with LPMS. RPMI is also known as basal media, and this is the media that is used to grow macrophages. LPS, which is lipopolysaccharide, when it's paired with RPMI, induces a pro-inflammatory phenotype. These two groups will be our negative controls, and this is because we want to ensure that macrophage-conditioned media is the only type of media that is having an effect on the cells, and that the RPMI and LPS are not having any effect on the cells. Moving on to part C of this figure, we can see that macrophage-conditioned media is isolated in order to collect the inflammatory secretome. The secretome produces pro-inflammatory markers, which is why this is our experimental group and the group that we are mainly testing to see if it is having an effect on the cells. And then in part D, we can see the timeline and overall layout of this study, which I explained previously. Another important thing to note is that all of these groups also contain chemically defined media, or CDM, which contains high glucose media, penicillin streptomycin, penstrep, which controls for bacterial infection, and L-proline and bovine serum albumin, BSA, which provide the necessary nutrients and proteins that allow the cells to grow. Moving on to the results of this study, we did biochemical quantification on the media that was collected from this study by doing a DNA assay using pico green and a GAG assay using dimethylmethylene blue. And then the GAG data was normalized to the DNA data. In observing the DNA data in part A, we can see that the macrophage-conditioned media group has a slightly lower DNA content than the chondrogenic baseline. However, all groups display an amount of DNA growth, which is what we would expect to see since DNA quantifies cell growth. In looking at the GAG assay, we can see that both the RPMI group as well as the macrophage-conditioned media group have a lower GAG content when compared to the chondrogenic baseline. However, when the GAG data is normalized to the DNA, we can see that there's no real difference amongst the different groups. In terms of the statistical significance of these results, a one-way ANOVA test was used because we have different groups, so we wanted to see what the difference was between these 5 groups. Moving on to the discussion and conclusion, our data suggests that macrophage-conditioned media is stalling chondrogenesis while also mimicking an osteoarthritic environment. Macrophage-conditioned media attempts to degrade the extracellular matrix when the cells are in pellet culture. However, this effect is counterbalanced by the chondrogenically differentiated HMSCs because they are immunomodulatory, which results in no observable levels of degeneration or chondrogenesis. In terms of our future directions and what we hope to continue to study, the current study used conditioned media from a murine cell line, but future studies will use a murine primary cell line with a greater potential for a pro-inflammatory phenotype. I would like to acknowledge the P.C. Rawson College of Engineering and Applied Science for providing me with the Claire Booth Luce Research Award, which is funding my research, and I would also like to thank the TGF Lab for supporting my research and providing me with this opportunity. Thank you.